Our next presenter is Kate Lubina. Kate is a PhD candidate at Rutgers School of Public Health in the epidemiology concentration. Her doctoral research focuses on investigating predictors and sources of PFAS exposure among U.S. volunteer firefighters enrolled in the FEMA-funded Firefighter Cancer Assessment and Prevention Study, or CAPS, at Rutgers. And in addition to this ERC pilot project award, Kate is also awaiting the funding for F31 predoctoral fellowship from NIEHS to support her doctoral research. And she also enjoys science communication and works on participant engagement in the CAPS study. So, Kate, welcome. It's all yours. Kate, are you
Oops,
talking?
there we go.
Oh,
Yeah, can you hear me okay now?
yes, we can hear
Okay,
you.
great. And now I've got control. Okay, so thank you so much for the introduction, Dr. Chu. My name is Kate Lubina. I'm a PhD candidate at Rutgers School of Public Health, and I'll be presenting our pilot project titled Exploring Exposure Sources of Per- and Polyfluoroalkyl Substances, or PFAS, among volunteer firefighters. I'd like to acknowledge and thank our funders, my mentors, and the CAP participants and research team who have made this work possible. We do not declare any conflicts of interest. So I'll present some background on PFAS and the connection to volunteer firefighters, our three specific aims of this project: methods, results, and some next steps we have planned. So PFAS are a class of chemicals that are made up of thousands of synthetic compounds that have been used in industrial processes and consumer products for over half a century due to their unique chemical properties. PFAS are chemicals of concern because of their ubiquity, their persistence in both people and the environment, and their associations with adverse health outcomes, some of which firefighters are at increased risk for. People are often exposed to PFAS by ingesting, inhaling, or absorbing PFAS through sources that can include contaminated food and water, consumer products, and for some groups, occupational exposures. Firefighters are one of these occupational groups, and there are additional pathways by which firefighters may be occupationally exposed to PFAS. These include combustion products, protective firefighting clothing, and historically a class of firefighting foams called AFFF. Several studies of career firefighters have found elevated PFAS concentrations compared with reference populations. However, there's a very limited literature on volunteer firefighters, despite the fact that volunteers make up approximately 2/3 of all US firefighters. So the aims of this project were to evaluate associations of volunteer firefighters' PFAS concentrations with demographic, health, and firefighting factors in the entire CAPS cohort of 553 participants. We also aimed to compare the serum PFAS concentrations in 213 New Jersey-based CAPS volunteer firefighters with a state-based comparison group, the New Jersey Biomonitoring Program, or NJBM, overall and by county. And finally, to explore the utility of expanding the number of PFAS analytes that we measure in a subsample of CAPS participants. I will focus on aim 1 first and then briefly discuss the results of aims 2 and 3 for the sake of time. So my work is conducted within the research infrastructure of the Firefighter Cancer Assessment and Prevention Study, or CAPS, which is a FEMA-funded study based at Rutgers that seeks to understand cancer in volunteer firefighters. CAPS is part of the National Firefighter Cancer Cohort Study, or FFCCS, and CAPS has adapted FFCCS protocols from the career context to the volunteer firefighter context. CAPS enrolled volunteer firefighters from 41 fire departments in 9 US states. And at enrollment, participants completed a survey where they self-reported sociodemographic, health, and occupational characteristics of interest. They also provided a blood sample from which colleagues at the New Jersey Department of Health quantified 12 PFAS compounds in serum using a CDC method. To accomplish the objective of AIM-1, we determined the prevalence or the detection percentage of the 12 PFAS using the lower limit of detection, or LOD, as our cut point. Measures below the LOD were imputed consistent with NHANES methodology. We further examined compounds that were detected in at least 75% of CAPS participant samples. We natural log transformed those PFAS measures to examine associations with predictors of interest in bivariate and multivariable analyses. Our predictors of interest for this analysis were captured by the CAPS enrollment survey. And our comparison group that I'll be highlighting is the non-Hispanic white adult male subset of 2017 to 2018 National Health and Nutrition Examination Survey, or NHANES. And NHANES is a nationally representative cross-sectional survey of the US non-institutionalized civilian population. So of the 12 compounds that were measured, 6 were detected in over 75% of CAPS participants. These were PFOA, PFOS, PFNA, PFHSX, PFDA, and PFUNA. We observed similar detection prevalence for these compounds between CAPS as well as NHANES. We calculated the geometric means and 95% confidence intervals for those 6 PFAS compounds that we detected in at least 75% of our participants. And each chemical here on the slide is represented by a different color. And we have populations of interest along the y-axis. So the non-Hispanic white adult males from 2017 to 2018 NHANES average serum PFAS concentrations are represented across the top row. All CAPS participants' averages and confidence intervals are on the second row. And in rows 3 and below are the average concentrations among CAPS participants according to the geographic regions that they are located in. And I'll highlight some additional points of interest here. So for most of the PFAS that we assessed when we compare CAPS and NHANES, the average levels were similar or lower among CAPS participants. And so we can see this when we take a closer look at PFUNA, which is in red, PFDA and PFHSX, which are in the blues, and PFOA in green. And we see quite a bit of geographic variation in the average levels of many of these compounds when we look at the regional subgroups as well. When we look at average PFNA levels, the CAPS participants have higher average levels compared to NHANES, so those first two comparisons. If we follow down this vertical pink line, which is the average level in NHANES, average concentrations in CAPS participants were higher, except for among firefighters from Kansas, Missouri, and Washington. And this is interesting to us because a few studies of career firefighters have also reported somewhat elevated average PFNA concentrations compared to reference populations. And the last thing that I'll point out on this slide is that we see average PFOS levels among NHANES participants that were about twice as high as those in CAPS participants. And so this brings me to a limitation of these data that I would like to highlight that has to do with the timing of when these measurements were done. So it's important to note that differences that we have observed may be due to temporal incongruities that can be attributed to when these two populations were sampled that we're comparing here. So CAPS biomonitoring was conducted between about 2 and 5 years later than the 2017 to 20 NHANES cycle, which are the most current PFAS data available from NHANES. Many of the compounds that we measured have a half-life of between 2 and 8 years in humans. And so as the figure here shows, we've observed time trends associated with changing population-wide exposures since NHANES began biomonitoring PFAS in 1999. And PFOA and PFOS were phased out of production in the United States in the early 2000s. And so we can see that the average levels of these chemicals have declined. So it's possible that some of the differences we're observing between CAPS and NHANES might be due to these time trends owing to the population level exposures. And so this adds a level of complexity when we're thinking about occupational versus non-occupational exposures to ubiquitous chemicals like PFAS. And we ran multivariable models for each of these 6 natural log transformed PFAS that we've been discussing. In the multivariable models, we observed associations between the concentrations of all the compounds we examined and geography. We observed positive associations between each of these 6 PFAS and age, being male and having 4 or more years of post-secondary education. We also found that responding to more than 20 monthly calls was positively associated with PFDEA concentrations, with weaker positive associations observed for PFNA, PFOS, and PFHSX. We saw particularly strong positive associations between being a career firefighter and PFOS and PFHSX levels. And we did not observe meaningful associations between PFAS concentrations and participants' race or ethnicity, years of firefighting experience, or a history of active duty military service. So this work adds to a limited body of research on volunteer firefighters who are an understudied occupation population. CAPS is a large geographically diverse cohort, and identifying sources of variation in PFAS exposures may point to exposure sources, and it's something that we plan to investigate further. And as for limitations, with cross-sectional data, we're not able to infer temporality in the potential source exposure relationship. And we just already have touched upon the temporality issue between our study population and our comparison population. So in AIM-2, we compared the average serum PFAS levels between New Jersey CAPS and a New Jersey-based reference group. And in AIM-3, we explored the utility of expanding the number of PFAS we measured in CAPS participants. The results of AIM-2 and 3 suggest that CAPS volunteer firefighters overall do not appear much more highly exposed than the US population or their surrounding communities. And we suggest trying to perform non-target analyses as opposed to expanded PFAS panels when exposures to novel compounds with shorter half-lives are suspected. So next, we plan to evaluate additional predictors of PFAS concentrations to attempt to further elucidate possible exposure sources. And we'll use 2017 to 2020 pre-pandemic NHANES data as a reference population once those PFAS data become available. I'll also be investigating the effects of PFAS exposure on blood lipid measures among CAPS volunteer firefighters overall and in the New Jersey subset. And to do this, we'll be using some additional analytic approaches, including some mixture modeling. And this work will be part of my doctoral dissertation. This pilot project generated data that informed feasibility and development of my F-31 submission, as well as my dissertation proposal. So thank you for your attention, and thanks so much to ERC for supporting this project, and I'm happy to take any questions.